Hi, this is Maggie Virus, and this is my Chapter 4 presentation talking about bacteria and all its functions. So first I want to talk about bacteria and what it does and what is inside of it. So bacteria are responsible for reproduction, they can function with metabolism, and they process nutrients. So all bacteria, they have cell membranes, cytoplasm, ribosomes, and chromosome, but there are certain bacteria that don't have certain things. So some bacteria can contain a cell wall, flagella, pili, fimbriae, and all the others included, but these are not found in all bacteria. So now I want to talk about the shapes of bacteria. They include the coccus, rod, and curved shaped. And the rod is sometimes called a bacillus, and curved is sometimes called a vibrio. So the coccus, these are spherical shapes. They look like balls, and this is like the top right picture. Whereas the rods, these are longer, more cylindrical shapes, but they're not always perfect. They can appear as drumstick shapes or like a club shape. And then there's also the coccobacillus. These are shorter and more plumped rods. And then there is also filamentous rods. These are longer and more thread-like. And they are kind of like a string, not as thick as a normal rod. And lastly, we have curved. These occur as a spirillum or a spirochete. The spirillum is a rigid helix, so it's not very flexible. It's a looser coil, whereas the spirochete, these are more flexible, and the helix is more tightly coiled. These are all examples of pleomorphism. This is when cells in the same species, they won't always appear the same. So though you have like the vibrios, there is also sub-shapes with like the spirillum and the spirochete. So I'm going to talk about the variety in cocci. So we have diplococci, which occurs in pairs of two. We have clusters like the staphylococci and micrococci. Staphylococci is like the bottom right shape. It's like a pyramid almost. It's an irregular cluster. Then we have chains. This is when many cells form together. There can be only a few, or there can even be up to 100. And then we have cubical packets. Like these are often, there's a Saccharina is a good example. This is when there's kind of like layers of cells on each other. So like the top example, there's a group of 4, and then I try to draw it behind it. There is supposed to be another group of 4 forming like a cube, a 2 by 2 by 2 cube. So now I'm going to talk about the variety in rods. So we have the Diplobacilli, which is in the main left red shape. It's when 2 rods are together. And then the Streptobacilli, this is a longer chain of this bacilli. And then we have palisades. These are still a longer chain. They can have several cells, but they're only partially attached on the ends. So bacteria live in biofilms. So biofilms, these are moist locations that have polysaccharides and glycoproteins. And cells will come here and they will multiply, and they secrete substances like DNA molecules or even signal receptors, which bring in more microbes to their community. So bacteria doesn't live as a single cell. Commonly, they live in biofilms. So quorum sensing, this is how the microbes will communicate to each other and allow for more growth within the community. And an important defining feature is aerobic and anaerobic microbes. Aerobic and anaerobic, we know aerobic is liking oxygen, it wants oxygen, and anaerobic doesn't. So aerobic microbes, they'll be found towards the surface of the cell where oxygen levels are the highest, and anaerobic will be down at the lower surface of the cell at the bottom where there isn't much oxygen present at all. And biofilm thickness, early biofilms will be very thin because they don't have many habitants in their community, whereas over time they'll get thicker and thicker as more cells are present. So now I'm going to talk about the external structures of bacteria. So one of the most important defining features is the flagella. This is how some bacterial cells will move around. They're often found in rod-shaped and spiral bacteria, and they have a filament. The filament is a helical structure. It's made up of proteins, and it is inserted into the hook. So the hook connects the filament to the basal body, And the basal body is basically the motor for the flagella. So it's like made up of a stack of rings, and it's anchored to the cytoplasmic membrane and the outer membrane. So we have different arrangements for the flagella. There are polar arrangements and peritrichous. For the polar arrangements, we have monotrichous, which is a one single flagellum, whereas lophotrichous, this is when there is many flagellum on either end. And then we have the peritrichous. So this is when flagella are randomly dispersed around the whole surface of a cell and kind of look like hairs coming out from it. So flagella move when they have a chemical stimulus. So there can either be positive stimulants, which are attractants, or negative stimulus, which are repellents. So when there's a positive stimulant, they want to go towards it. They project themselves towards the positive stimulus. Whereas negative, this is when they want to get away from it. It's a harmful chemical. They don't want anywhere near it. And this is an example of chemotaxis. So chemotaxis, chemical stimulant. It's when they move because of some kind of signal that tells them to either go towards it or away from it. So flagella, they also have different strategies in their movement. They have a run, which goes straight towards the stimulant. It goes counterclockwise. It rotates counterclockwise to swim directly towards the stimulant in a linear direction. Then there's a tumble. This is something that interrupts the runs. It makes the flagellum reverse and change its course. And then we have a random walk. This is a combination of the two, so it alternates between a run and a tumble. And then lastly, we have the biased random walk. This is when bacterium will slow down the amount of tumbles it does, allowing for a longer run time, but still tumbling sometimes. Then we also have periplasmic flagella. These are found internally, and these are internal flagellum. So they help them move, but they're located not in the same place that flagella usually are. So these periplasmic flagella, they're also called axial filaments, and they're located within the cell wall and the cytoplasmic membrane. So now we're going to talk about the external structures of bacteria more. These aren't involved in motility. So there's the fimbriae, the pili, and nanowires. Fimbriae and pili, they help attach to stuff, but fimbriae are more attaches to bacteria and surfaces, and they're like short hair bristle like fibers, whereas the pili, they are longer tubes and they connect two bacterial cells together. And this occurs usually during conjugation. And this is when the two bacteria will share DNA. But this occurs in gram-negative bacteria. But in gram-positive bacteria, they use aggregation proteins instead of pili. And then last we have nanowires. And they look like hair, and they come off of the cytoplasmic membrane, and they're used to share electrons and nutrients. So now we're going to talk about the layers that help protect the bacteria. So we have the glycocalyx, which this is a coating around the cell, and it's made up of polysaccharide units and sometimes a little bit of protein, and it helps protect the bacteria cell and adhere to its environment, surfaces in the environment. And the thickness of it varies, so we have the slime layer. This is a looser coating, and it also helps with preventing water loss, whereas the capsule is a thicker, denser coating, and it provides more protection. So the glycocalyx and the S layer, they can either be used individually or together. So the S layer is produced when it's in a harmful environment and the bacteria thinks that they need more protection. So it helps protect the cell, and it also helps attach to surfaces and make colonies bigger. So now we're going to talk about the cell envelope. So the cell envelope is a boundary of bacterium. It helps protect it, and whether it's gram-negative or gram-positive, it can have 2 to 3 layers. So this includes the cell wall, cytoplasmic membrane, and outer membrane. Okay, so I'm going to talk about the gram-positive and gram-negative. So gram-positive is the one with 2 layers. So it has a thick peptidoglycan wall, and it is probably their defining feature. So it makes them better, stronger, and can resist osmotic pressure. And then just beneath it is the cytoplasmic membrane, and this regulates what enters and leaves the cell. Then we have gram-negative bacteria. This has an outer membrane, so now it has 3 layers, unlike the gram-positive, which only has 2. So they have the outer membrane. This adds protection, and it also makes it harder for antibiotics to get to them. And then we also have their peptoglycan wall, the thin cell wall, and it's much thinner compared to gram-positive bacteria. And then lastly, we have the cytoplasmic membrane. This is the innermost layer. So bacterial cell wall, it determines its function and shape, and it's critical for stability. So the cell wall prevents the cell from collapsing when there's changes in the environment or changes in osmotic pressure, and its key component is peptoglycan. And peptoglycan is like a mesh-like structure, chains tied together by short protein fragments. And a common example of bacteria that doesn't have a cell wall, so sometimes they don't have a cell wall, not always. So pleomorphic mycoplasma. You've heard of myco or walking pneumonia. This is an example of this bacteria. It gets into your lungs and causes them to get sick, giving them mycoplasma or walking pneumonia. So now we're going to talk about their cell wall. So gram-positive cell wall is very thick and strong because of its many layers, and it contains different acids and special molecules like teichoic acid and lipoteichoic acid. It helps anchor the wall to the cell membrane, and it contributes to its strength and function. So when using the differential staining method, gram staining, this bacteria will remain the original color of the stain and appear purple. So this indicates that it's gram-positive. Then we have the gram-negative cell wall. This is a thin layer of peptoglycan. It makes it more flexible, but it also makes it more fragile and sensitive to lysis. So when you do gram staining, the differential staining technique, it appears pink or red, and it indicates that it's negative. So now we're going to talk about the gram-negative outer membrane. So the gram-negative outer membrane, they have an additional outer membrane compared to the gram-positive. So this is the upper layer made up of lipopolysaccharides, or it's also called LPS, and these contain endotoxins, and these help trigger immune responses like stimulating a fever or medical shock like in infections like meningitis. So the innermost layer of this is made up of phospholipids, and it also has porrin proteins. These, I don't think they're on the image, but these are helping control what substance can enter the cell, and it maintains how selective it is. So now I'm going to talk about the bacterial cytoplasm. So the bacterial cytoplasm, this is the internal fluid found within the cell, and it surrounds and provides a nutrient processing like synthesis, and it regulates the transport of nutrients into the cell, and it also regulates the waste going out of the cell. So this is made up of mostly water. It is the main component. It takes up about 70% of the cytoplasm, and it acts as a solvent. So it helps dissolve nutrients, like when amino acids or sugars or salts, it helps to dissolve them. And then within the cytoplasm, you'll find structures like chromatin, granules, cytoskeletal fibers, or ribosomes. So I finally want to talk about the components within the cytoplasm. This includes genetic material like DNA. It's inside the cytoplasm, but it's found aggregated within the nucleoid. So the DNA is a circular, it has no loose ends, which is different from eukaryotes. You know, there's free ends on the DNA helix. This is different. It's in a complete circle. And ribosomes, these are made up of RNA, rRNA, and proteins. So they float freely in the cytoplasm, or they can attach to the cytoplasmic membrane. So multiple ribosomes, they can come together and they can form chains called polysomes, and this helps make protein production more efficient. Finally, I want to talk about the cytoskeletal fibers. These are helical ribbons. They're shaped like helixes, and they surround the cell underneath the membrane. So their fibers, they resemble actin and tubulin, and they contribute to maintaining the cell shape and by affecting the peptoglycan production. So this is cytoskeleton helps alter the shape. And that is all for my presentation. Thank you for listening.